Okay, our next speaker is all over the internet. All you have to do is put in the letters MTHFR and Ben Lynch will come up everywhere. Matter of fact, when I started doing some research in this particular area, really all I could find was that was credible was stuff from Ben Lynch. And so, thanks to a sponsor of SpectraCell, and please go visit their booth, we were able to actually get Dr. Lynch to come speak for us. So I would like to turn it over now to Dr. Ben Lynch.
All right, thank you. All over the internet. I don't know what that's a good thing or a bad thing. No, just don't search Ben Lynch in forum. All right, so up, down, and laser. Okay, so how many of you guys and gals use nitrous oxide in your clinics? All right, how many of you have taken it personally from being on the other side of it? Okay, well, you're all on the other side of it because it's in your clinic. And how many of you have kind of a little bit too drowsy, a little slow to come back to your normal self after you've used it? Oh, you guys. All right. Okay. Then you all have beautiful genetics and good epigenetics, so that's good. So I am pointing this, I assume, at the... It's not advancing. There it goes. Okay, got it. So the statement here at the bottom that clinicians are central to helping consumer patients utilize genomic information to make health decisions, that's a quote from the New England Journal of Medicine, and I did not write that article, okay? But that is a very useful statement because patients are coming to you highly, highly educated and sometimes grossly misinformed about how their genetics are affecting them, okay? But you're going to soon learn that MTHFR is definitely affecting their ability to eliminate nitrous oxide, or at least it's doing a dysfunction to their methylation pathway. Standard disclaimer disclosures. And this is a great article I'd like you guys to read. So sometimes I have slides in here, not for me to read, but for you to read on your own time and flying back on the airplane or what have you. But I am a big, big fan of nutrition. I am, after all, a naturopathic physician, graduate from Bastyr University in 2007. And I got my cell molecular biology degree from University of Washington back in '96. And nutrition is one of the primary environmental exposures that we're all exposed to. And we all know that our food is pretty well adulterated now, GMOs, pesticides, herbicides, and whatever else that's affecting it. And that food is then leading us to things and having issues with our patients' teeth, or various cardiovascular disorders, or what have you. So I think food is most important to start with. So if you look at this diagram, so epigenetics, right? We all know epigenetics? Raise your hand. Okay. Epigenetics is basically how our genes are told what to do. So we all have our genes as the computers. So we have the computers in our system, but the things that affect our genes is the epigenetics. So food, environment, water, drugs, prenatal care of your mother, all these things affect how your genes function. Your perception of the environment, taught by Bruce Lipton, fantastic as well. So drugs is a big player in how your genes function or don't function because they affect the epigenetics of your gene function, your phenotype per se. So we're going to be focusing on drugs because we're going to be talking about nitrous oxide and how it can affect your MTHFR enzyme and your methylation pathway and other things as well. And so all these things contribute to a lower capacity to do work, which is methylation. And then you get damaged mitochondrial and nuclear DNA. And then that leads to gene instability, altered gene expression, and a mess, pathologies. So methylation is simple. It's basically just a carbon and 3 hydrogens, and it just goes around and attaches to different things. And when it attaches to certain things, it changes the function. It turns things on, it turns things off, it transforms into less toxic or more toxic, depending on what it is. So, and it is a primary biochemical function that is happening probably 1 trillion times a second in every single cell. So let's look at uracil. Uracil is an RNA base. If our methylation and our folate metabolism is not working, we're going to be incorporating uracil, a RNA base, into our DNA. That's not good. We do have repair mechanisms. Our body knows how to restore that, so it goes and it splices out the uracil and it will put in a thymine. The problem is, if that keeps happening due to methylation deficiencies or folate deficiencies, that continued splicing is going to cause an instability of that gene, and it will start breaking. And if a gene starts breaking, it can't be transcribed, the end product cannot be made very effectively, and then the rest of your life, you're not going to be making the end product which you need. And another one is arsenic, which is everywhere. Brown rice syrup is commonly used as a sweetener for things, very high in arsenic at times. Rice, chicken are fed antibiotics laced with arsenic. So these chickens are eating, we just had chicken for lunch, some of us. So these chickens are fed these antibiotics all throughout their life because they're living in these horrible environments. So they're consuming that arsenic all throughout their life. They're building it up, because I don't think their methylation capacity in that environment is very good. And then we consume that chicken. And then we drink the water, which our former president has increased the safe levels of arsenic in our drinking water. So, and then we've got it in our chlorellas and our kelps. So arsenic is very, very pervasive in the environment. And you can see here that it requires glutathione. GSH is reduced glutathione. It requires glutathione and SAMe in order to get this out of here. So if you're just taking glutathione and thinking you're getting rid of your arsenic, you're not. You need adequate methylation as well. So basic functions of methylation, there's many. I don't want to get into this. Some of my slides are reference slides for future. And can you guys read the stuff on your slide? Is it big enough? Yes? Okay, good. If it's not, I'm happy to, I have a download link which I'll provide the organizer of this event so you guys can download my entire PowerPoint presentation where it's big because I have some diagrams in here that I want you guys to utilize. But basically, functions of methylation, it's biotransformation, regulating. I think of it as a light switch that's sitting on the wall. You have the ability back there to go turn this light off or on, but you need to actually physically go and do it. There's no automatic sensor. You need to physically touch it. And that physical touching for turning things on and off is a carbon and 3 hydrogens. So if I'm the methyl group, I walk over there, I flip that light switch on. The gene turns on, and I can also go there and turn it off. Okay? So neurotransmitter production. Your patients come in stressed out, they're scared of hell of dentists. You give them the nitrous oxide to calm them down so you can work on them and get them out of there. But you're going to learn that nitrous oxide is going to actually make their pain worse. And then they're going to be home at pain, and they're going to be associating it back to you guys as the instigator of that pain. So, because dopamine increases pain. So cell membranes are a huge consumer of methylation as well. Methyl donors, B12, folates, methionine, protein. So protein is your biggest contributor of methionine. Choline comes from meat. So vegans and vegetarians are going to be low in meat. They're also going to be low in methylcobalamin. Glycine and serine, they're pretty pervasive, but also in proteins. TMG comes from choline. DMG is a metabolite of DMG, and we'll get into all those. But I just listed them there for future reference. So let's look at this slide here. So everything I just mentioned, here's MTHFR down here. That's the gene I started with. I have a whole website dedicated to it, mthfr.net. There's a video on the home screen of mthfr.net. You can click that and play, and I think it's an 80 or 90-minute video. It's free. You can learn about it. But this is basically a complex look and a one-dimensional look of a very complicated pathway, but gets messed up from nitrous oxide. So these two areas right here, MTHFR and methionine synthase, are key players in how methylation works. And this is the methionine cycle. Okay, and the methionine cycle produces SAM, which then goes and it contributes the methyl group to all the methyltransferases throughout the body. And we'll get into detail. I'm just giving you guys a bit of brief biochemical stuff here. And you can see down here, there's a little diagram. Promoters are orange, inhibitors are purple. And there's nitrous oxide as an inhibitor of methionine synthase. And so there are issues with that, which we'll get into detail. All these yellow underlines is how nutrition impacts your patients and yourself and your own methylation pathway. So let food be thy medicine. If you're eating right and you're eating a comprehensive diet and you're not eating adulterated foods by our food industry, and you're doing as good a job as you can and you're absorbing those nutrients, then your methylation capacity is probably pretty good if you don't have all these things inhibiting it. And there's numerous things that inhibit your methylation. So nitrous oxide, acetaldehyde comes from alcohol, yeast overgrowth, lead, mercury, LPS, found in bacteria cell walls, carbon tetrachloride, Hep B, Hep C, TNF alpha is an inhibitor, and it's actually an inhibitor of methionine synthase too. So if your patients are inflamed, their methylation is not working very well. And SAM, a remarkably versatile molecule, is said to be second only to ATP in the number of functions it does. That's huge. Our body produces ATP, it produces ATP in weight that's equivalent to your body weight every single day. That's a lot of ATP. And 40% of your ATP at rest is utilized just to move your electrolytes in and out of the cell. So there's a lot of ATP utilization just passively to move your potassium into your cell, the sodium out, and to get the magnesium in. Okay, so that's and those are ATP pumps that do that. Now SAM does over 200 reactions, and they're very, very significant. And I don't go into all the different reactions here, but I will say that it's a very important point, is 70% of your body's utilization of SAMe is made to produce phosphatidylcholine and creatine. So if your cell membranes are not doing very well, you can automatically assume that your methylation is hampered, right? Because phosphatidylcholine is a key component of our cell membranes. So phosphatidylcholine is a critical nutrient, and if you supply phosphatidylcholine to your patient, you are going to preserve their methylation, because that frees up 70% of the SAMe to do other things, like make neurotransmitters, get rid of histamine, and so on. So here's your SAMe, makes methyltransferases. And this is an interesting point. Look what promotes methylation. Glucocorticoids. What's a primary glucocorticoid in humans? Cortisol. Stress. So you come to the dentist, you're stressed out, possibly. I don't know of many kids who love going to the dentist. I know one, and she actually wants to be a dentist already, and she's in my son's class, and he's beside himself because he can't stand dentists. But to his own fault. He needs to brush his teeth better. But stress promotes methylation. So that means that they're utilizing the nutrients required to run all this. So stress is catabolic, we know that. And it's catabolic because it's using up all these nutrients. Part of the reason. Multiple reasons, but that's one. And this is a look of how SAMe is utilized by the body. And I just told you that the major reactions involved in transmethylation, which is the methionine cycle, which I just showed you. The primary one is GAMT to form creatine and PEMT to form phosphatidylcholine. And I can tell you right now that both of these are polymorphic in the population. GAMT meaning that there are quite a few kids and adults who have a decreased capacity in this gene right here. That means their ability to form creatine is reduced. And my wife has a significant PEMT SNP, so she's not able to produce her phosphatidylcholine very well. And she also has numerous other genetic problems. We're all mutants, all of us. So we all have some degree of SNPs in us, okay, to some degree. And so understanding what you're susceptible to is super important. And so if you run a 23andMe test and you start looking at the raw data and you say, look, I have this GMT and PMT, that's useful to know, isn't it? You know that you're susceptible to having cell membrane damage. So you need to support that. If you see low creatine on a lab, then you might be suspecting also low methylation. And I can tell you too, if you're working with kids, how many of you guys work with kids? Okay. How many of patients do you see are slow to speak? Anybody? Okay. So GMT, even Children's Hospital knows to screen for this gene for slow verbal development in kids. So you support them with creatine, then they are able to support their methylation to actually start speaking again. So GMT is a known polymorphic, GMT polymorphism is known to reduce the ability for kids to speak. A little sideline from nitrous oxide. I'm just passionate about some of these things since you guys see kids. So these are the things I drop for fun. I love biochemistry. That was my favorite class in high school. So SAM formation is complicated, but at the same time, it's a beautiful thing to watch. And the cool thing about these diagrams is you don't have to learn it. You can just look at these diagrams and you can understand that, God, you know, my patient does have mercury amalgams and they have yeast overgrowth, and I got to give them nitrous oxide, and they also have possibly some, you know, Hep B or Hep C, or they're inflamed in TNF alpha, so you can automatically know and look at this and say, God, you've got a block here and here. So that means you're reliant here to back that up. Okay, so it takes time. You look at this one arrow at a time, and in time, it will make sense to you. And after a while, you'll be able to see this in your head, and when you're taking a history from your patient, you'll automatically say, God, your methionine synthase is blocked. We need to support that. And I'm going to teach you here today how to do that. Here's MTHFR. Very, very centralized. So if your MTHFR enzyme is screwed up like mine, then your ability to produce this end product, 5-methyltetrahydrofolate, is significantly reduced. But that does not mean if you have MTHFR, like I do and over 50% of the population to some degree, have a decreased capacity to do that. If you eat your leafy greens, so I just had some leafy greens today, I got some methylfolate from that. So if your diet is and you're consuming your raw leafy greens, you might be fine. You might not need extra methylfolate. And so that flows right into the methionine synthase enzyme, and that starts driving all this. This is a tag team. Methylcobalamin and methylfolate work in unison to support the methionine synthase, which takes your homocysteine and it recycles back into methionine to produce the body's number 2 important compound in the body, SAMe, number 1 ATP. You guys following me with that?
So what you're saying is you can't produce methionine yet?
Methionine, that comes from protein. So in the beginning of the presentation, I underlined with orange in the slide a lot of these different things. And methionine was underlined as well. Methionine comes from your protein. And that can be from beans, rice, nuts, seeds, what have you. So from your diet, you get methylcobalamin if you're eating red meat. You're getting methylfolate from your leafy greens. You're getting your methionine from protein. You're getting your magnesium from leafy greens and lots of other things, nuts, seeds. Choline, you're getting from meat. Serine and cysteine, you're also getting from proteins. So a lot of this is supported by your diet. Yes?
So I've always heard to give them methionine if you give them nitrous.
Give them methionine if you give them nitrous? Yes. So, and we'll get into that in the second talk. So, nitrous oxide, is it bad? So, it's employed in dentistry, obviously, for the primary purpose of reducing anxiety, and I'd also say to make it feasible for you guys to do work on your patient, right? I mean, if they're squirreling all around on the table, it's kind of hard to do work. So, and that's a pretty significant number of people who try to avoid dentistry. So if we can reduce fear in your potential patient, that means you've got 20 to 40 million more patients. That's a pretty significant number. So, and I pulled this slide off of some other person's presentation, and he was a dentist. I should have cited it. So, this is from the Lancet. The effect is well known among anesthesiologists or anesthetists that nitrous oxide irreversibly inactivates cobalamin 1, the active form of vitamin B12, which is essential for the methionine synthase enzyme, which I just showed you guys, which was an MTR in that pathway. So the methionine synthase activity, and it converts it to cobalamin 2. And I will show you that cobalamin 2 is a problem, but the body can fix that problem. And most of my slides have citations. I'm not just making this stuff up. So, here's MTR, methionine synthase. Now, here's the effect of nitrous oxide. It's going to blow out and oxidize cobalamin 1. So if it blows up cobalamin 1, it's producing cobalamin 2. That increases. Okay? So what's the body do? Well, when that happens, it produces superoxide right here. You guys know what superoxide is? Very, very pro-oxidant, very damaging, super damaging, which is also a very polymorphic gene in the body, which my wife and a lot of our children have as well. So superoxide dismutase is a gene which eliminates your superoxide into hydrogen peroxide. What eliminates hydrogen peroxide? Anybody? Catalase, what else? Glutathione. Thank you. Good old Chris, he's figured that out. Okay, so glutathione and catalase get rid of superoxide, or of hydrogen peroxide. Then, so that increases. Now, if your cobalamin 2 increases, your methionine reductase is undergoing more work. And it has to do more work, and it has to use SAMe, riboflavin, and niacin to convert this cobalamin 2 back into methylcobalamin. But if you're inhibiting your methionine synthase, or you have a lack of methylfolate, or your patient has a lack of methylcobalamin, your ability to have SAM available is reduced. You can back it up from the choline pathway through BHMT. Problem is, this gene right here, that produces that BHMT that enzyme, is only typically found in the liver and the kidneys. Whereas this pathway is everywhere, this one's only in the liver and kidneys. So the brain, ovaries, testes, other tissues are going to be suffering, and the homocysteine in that particular tissue is going to elevate. So as your homocysteine increases because of this blockage, your BHMT will increase. But if your patient is a vegan or vegetarian, they don't have adequate choline, this backup pathway is not going to occur. So then their homocysteine is going to increase even more. So this is a shortcut route to convert your homocysteine back into methionine. So when you did state that one of the things that you can do to relieve the damage done by nitrous oxide in your patient is to give methionine, well, that's true, but if the root cause of the problem is oxidized cobalamin, giving that is not the answer. It can help, but it's not the actual solution. And so what do you see on your labs too? You can actually see an elevated serum folate in your lab. You can see an elevated serum cobalamin. Is that useful? No. because now you have oxidized cobalamin. It's not useful, it's not functional to the patient, but you order the lab test, it shows that their cobalamin and their folates are fine. Well, their 5-methyl tetrahydrofolate is elevated because it can't get in here to do work, because it needs methylcobalamin to do the job, and it doesn't have it. So just because you have methylfolate here, it needs methylcobalamin to do work to support it. It takes 2 of these nutrients to generate methionine synthase to convert homocysteine to methionine. Does that make sense? You can't just have methylfolate to cruise up into here to convert your homocysteine to methionine. You also need the methylcobalamin, but your methylcobalamin, if your patient is glutathione deficient, SAM, riboflavin, or niacin deficient, then this is not getting regenerated. That makes sense. And a lot of our patients are deficient in B12, especially the vegans and vegetarians or those on antacids. And you've probably seen a lot of reflux in your patients as well. I'm not going to go there. So I found this, and I was like, oh my God, that's real proof that shows how significant nitrous oxide is on methionine synthase activity. Now remember, methionine synthase activity is right here, okay? Right here. So during treatment with nitrous oxide, there is a 20% function left in the brain, okay? In the liver, there's only 5%. After 2 days, the brain recovers back to 95%. The liver is still struggling around 55%. Now, it's because the liver performs about 90% of the methylation reactions in the body. So the liver does most of the brunt of methylation. So, and here's the source for that. I was like, God, that's significant. But then I read the study in full, and it talked about it was on rats, and it was using like 90% of nitrous oxide and very little oxygen. You guys don't do that. So this is not really relevant to what you guys are doing. So I said, I need to find some human studies. Human studies, looked at 70%. That's pretty high, right? You guys don't use 70%. I mean, how many of you guys use about 50, 60% for an adult? Okay. Any of you use 70? A few. 65. Okay. Okay. So 30 to 290 minutes. We're probably talking surgery of some sort, not dentistry. So half-life methionine inactivation is 46 minutes. So the liver was reduced to 45% after 45 minutes with a 70% utilization of nitrous. Now, occupational nitrous, which you all have, you have increased oxidative damage, potentially, because your B12 is oxidized. If your B12 is oxidized, your methylation is going to be potentially hampered. And then we have 60% in a 3-month-old death. And again, 45 minutes, and then they had probably go in again. That's pretty significant, and we don't know what happened if that was a B12 deficiency or not. And then we had progressive numbness and ataxia that was reversed with B12 injections. And I don't know the percentage utilized here, but that was just dental surgery, so probably what you guys used. So mechanisms of toxicology of nitrous, we've already gone over this. It inactivates B12, but this is just another paper to reinforce what I said, because you can always find a paper to support your claim, right? So I want to find multiple papers to support my thoughts on this, to show you that this indeed is a problem. You guys use 50 to 60% of nitrous in your patients. Yes, it is needed at times, and I will show you how you can support your patients so you can continue using it. So I'm all about optimizing your patient, so in the patient's experience, and your practice as well. So I'm not here to bash nitric oxide. I'm showing you how to be proactive in identifying your at-risk population and to reduce the side effects and to optimize the autistic population that we're seeing increase on a grand scale. So Dr. Richard Deeth, he's done a lot of work with dopamine receptors and in the autism community. He's a leading researcher, and he was kind enough to let me use this slide. And this is really key. And so methionine synthase oxidation, and it's very sensitive to oxidative stress, and I'll show you why that is. It's actually protected in this mechanism by the body, and I'll get into that in a second. So oxidation of cobalamin, which we talked about already, but that only happens about 1 turn out of every 100. So you have your methylcobalamin coming around and doing the work, and under normal situations, that cobalamin does not get oxidized. But if you're utilizing nitrous oxide, it's going to oxidize it, okay? And it needs these nutrients of SAMe, methylcobalamin, riboflavin, and niacin to recycle that oxidized cobalamin. And then it needs glutathione to repair it. So it makes that enzyme dependent upon methylcobalamin and glutathione. 2 major deficiencies in the autistic population, the elderly and the chronic diseases. The enzyme will stay off until there's enough glutathione. Mercury amalgams. Glutathione is going to be hampered, isn't it? We've already pointed this out. This is a building. This is complicated, but the more you hear it, the more you apply it. In the second talk, you get another hour and a half of me. I don't know who suckered you guys into that, but you will start seeing this, and it's going to start sticking in your head. So if this is the problem, then glutathione is a problem, but Dr. Deeth is assuming that your patient has the ability to get rid of superoxide. They may not. And you can tell that by ordering RBC zinc and RBC manganese. And we get into that later. If they don't have adequate manganese or zinc, or possibly even copper, then their ability to convert superoxide into hydrogen peroxide isn't going to occur. So, yeah. So look here. Here's your catalase, which was talked about to you guys before. Here's your hydrogen peroxide. So see, we talked about how superoxide is needed to process, superoxide dismutase is needed to process this superoxide right here. And this is just an example of how reactive oxygen species are processed by us. And the cofactor in green is copper, zinc, and manganese. So superoxide goes through a functioning SOD enzyme, not a polymorphic one, which is actually pretty high in the population, cruises down here to hydrogen peroxide. You need catalase, which requires iron. Serum ferritin is pretty low in women. So you got to check your serum ferritin. And it can also go through glutathione peroxidase. So hydrogen peroxide goes through that route, and that's the 2 pathways for that. Now, here's just another look of how the pathway connects everything. So here's your superoxide. It's here. It shows you how it's metabolized. And then here's your hydrogen peroxide. It shows you how that's metabolized. Make sense? So who's at risk? You are. You guys are dealing with nitrous oxide every day, possibly, in your clinic for multiple hours. Okay, there's probably checks and balances that you have to do, that your tank is measured, and there's procedures and all that. But you are probably getting some low-dose exposure to nitrous oxide on a continuous basis. Would you fault me in that statement? Is that wrong? No? Okay. Yeah, okay. All right. So 30,000 dentists and an equal number of assistants have neurological complaints who have worked with nitrous oxide. Numbness, tingling, muscle weakness, and the 4-fold greater complaints for non-aesthetic exposed dentists. And I encourage you to grab these papers and to read them in full. And this one as well, you can grab. So by the far the largest group, okay, so cobalamin, nitrous oxide, well, that was addiction. So the commonest early symptom was numbness, okay? So again, I'm not up here to read to you. You guys have the ability. And this was talking about a 50% oxygen mixture. So we've talked about the objective of nitrous oxide and oxygen mix. The main reason is to reduce anxiety in your patient so you can work and be efficient and get them out and get another patient in so you guys can make some money. You can't be wrestling with one patient all day long. It's not practical. Now, this statement here, it raises the pain reaction threshold. Well, it is an NMDA antagonist. Nitrous oxide does reduce glutamate activity by reducing NMDA activity. Now, the problem is that's during the very moment. But since it does disturb methylation, it is going to amplify pain in your patient post-treatment. And I'll show you why later, because it's going to elevate dopamine. And dopamine requires SAMe to be metabolized. And if you have higher levels of dopamine, your pain is more intolerant. So people, if you ask your patient, are you more susceptible to pain, yes or no? Say, yeah, I'm actually very sensitive to pain. Or you can ask them, so if you're not susceptible to pain, or you're not a crybaby with it, doc, you do whatever you want. I don't even need any nitrous or anesthesia. Their dopamine levels are probably lower than the patient who was more susceptible. Does that make sense? So this lists, and we're going to get into this pretty in depth, reviewing the patient's histories, obviously adverse drug reactions, diseases and disorders, but they don't label them, which is an issue. I'll get into that. And contraindications, severe emotional disturbances. That's interesting, because if your methylation is disturbed, your neurotransmitter function is going to be disturbed. And they know that. First trimester of pregnancy. And then you'll see somewhere else where that's a suggested contraindication. I say it's an absolute contraindication. You don't want to have methionine synthase blocked in a woman who's in her first trimester, because the methylation cycle is cruising, because that baby is developing rapidly. And if you block methylation, the cellular division and everything else is not going to be occurring. And neural tube defects are going to be at risk, and Down syndrome and everything else. Methylene tetrahydrofolate reductase deficiency. What's that? What's the acronym for that? MTHFR. Cobalamin deficiency. Okay. So there's numerous mechanisms of neurotoxicity, because it is an NMDA receptor antagonist, and or actually I don't know if it's receptor antagonist, but it's an antagonistic to NMDA. Vitamin B12 deficient patients, elevated homocysteine, you can screen for that, but that's not very effective because there's multiple routes of homocysteine metabolism. I have issues with homocysteine being the only test utilized for that. There's a wide range of patients to whom nitrous may have some form of toxicity. Now, you might not see it immediately. There's a delayed response. Remember how that previous slide I had up there showed that after 2 days, the liver's ability to methylate was still compromised, correct? So you can still see some issues in your patients after they leave your clinic. So here it talks about a case study, details a patient who presented with weakness in their lower limbs as well as peripheral numbness. The patient was deficient in B12, and then they found out that same person was exposed to nitrous oxide for 2 dental surgeries 2 to 3 months previously. So again, a time delay, and multiple treatments, and B12 deficient. So again, it's understanding your patient population, understanding your patient, and understanding what else is going on. So team care with that particular patient you're working on is important, requesting charts, or just doing a screening. And again, here's another paper. Here's the source. It's hard to see, but there it is. You can download the full PDF. So human data suggests that 70% nitrous oxide results in a 50% reduction in methionine synthase activity. That's basically saying that there's a 50% ability for your patient to do methylation in every single cell of their body, except their liver and kidneys, if they have adequate choline on board. So you've reduced your patients at a 70% nitrous oxide utilization, you've reduced it down their methylation by 50% within 46 to 90 minutes. With no activity, again, being delayed after 200 minutes. So there's no activity left in methionine synthase after 200 minutes of use, sorry. And recovery appears to occur within 3 to 4 days. And here are some risk factors of B12 and folate deficiency. So you guys probably, can you even read that on your slides, or is it too little? Can you read it? Okay. Again, I'll send the organizer the full slide so you can see this more clearly. And now it's talking about SNPs and the MTHFR gene. So far more relevant, however, are certain fairly recently described, it's not very recent, but never mind, MTHFR polymorphisms. The 677 variant is the more serious one. And 1298 isn't too bad unless it's combined with 677, or unless it's combined with 677. I don't want to get too much detail on this. Basically, 677 and 1298, that's a position in the gene. That's where in that gene, which is like, I don't know, a few thousand base pairs long, I'm guessing. So at 677th position, there's a switch from cytosine to thymidine. That's all that means. But that's not relevant to dentistry. Just you need to screen for it and understand it. So having two polymorphisms, two 677s or two 1298s, or a combination of one of each, such as myself, it may lead to higher baseline plasma homocysteine levels and greater post-operative increase after nitrous oxide exposure compared to the normal wild-type patient. Wild-type meaning the MTHFR gene is not, there's no base pair changes. So who's at risk for MTHFR? So if you look here, now this is homozygous 677, which is pretty significant. Homozygous 677, they have about a 30% capacity to produce methylfolate. Only 30% compared to the wild type. But if they're eating their leafy greens, that's not a problem. Okay? They're eating their leafy greens, it's not a problem. So nearly 40% of Mexicans have this. Italians, Chinese, Hispanics, and whites. Okay? So understanding your patient population by ethnicity is important. Now that's 2. 1 copy is about half. So every other patient that walks into your office, and I've done probably well over 1,000 MTHFR tests, and I can vouch for this. If you guys start testing MTHFR, requesting that your patient's doctor test for MTHFR or themselves through 23andMe, which is what I recommend because it's inexpensive, but it's not the full picture. You can go, well, 23andMe is a full picture, but sometimes that opens a can of worms, so I would use SpectraCell for ordering MTHFR. I would not use LabCorp. They can come back and charge your patient multiple thousands of dollars for one simple genetic test. So LabCorp requests can be significant problems. SpectraCell will not do that to you or your patient. Again, hammering this. So this is something that we talked about, an absolute contraindication, a known deficiency of an enzyme or substrate in the methionine synthase pathway. What's a known substrate in methionine synthase pathway? Come on, tell me. Something. What's the substrate? It's an ingredient, right? If you're trying to make cakes, you need flour, sugar, eggs. So if methionine synthase, the MTR enzyme, needs to do work, what does it need to do work? What 2 nutrients? Thank you. What form of B12? Methylcobalamin. And what form of folate? MTHFR folate. Yeah, the mother effer folate? Yeah, yeah, yeah. It needs the methylfolate. If you get folic acid, I'll show you the folate metabolism quickly. It's stupid. It's stupid to use folic acid. Now, see, it said relative contraindications first trimester. That's ridiculous. So I moved it up there because I can do that with red arrows. So at-risk population for nitrous, again, this is a reference slide for you. I'm not going to go down through this, but basically all I did was I looked at the substrates of methionine synthase. So I looked at the things that your methylation cycle needs in order to do work. And I listed them. That's all I did. And then at the same time, I looked at what inhibits the methionine synthase enzyme or methylation in general. So I did two things. Things that interrupt it, things that support it. You need to know both, right? So, methylfolate, cobalamin, riboflavin, SAM, niacin, methylcobalamin here. Oops. Now we also listed the things that interrupt it. LPS, hepatitis, TNF-alpha, inflammation, IL-6. And then look at what interferes with methyltransferases. Bisphenol A, green tea extract, which I think I put in there wrong. Polyphenols, chlorogenic acid, which is promoted like crazy for weight loss. It's ridiculous. That shuts down methylation. So you don't have to remember all the nutrients that are needed which affect your methylation or the things that inhibit it, okay? So which support or inhibit, it's all here for you. And this is your main area of concern. And I'm going to very, very much simplify this at the end for you. I promise. Because people come to my conference and say, Guy, Dr. Lynch, that was great information, but I felt like I was drinking out of a fire hose. So I'll get it down to a trickle at the end. I just want to show you the complexity and how everything works and then how to fix it at the end. That's how medical school is, right? They shove the anatomy, physiology, biochem at you, and you're like, God, that's crazy. And then the last couple of years of how to fix everything. So it's kind of like today, too. So things which deplete B12. So exposures to chemicals, and there's numerous of those. So the key point with this slide is if you have depletion of glutathione, then you are going to have problems with physiological levels of B12. So extreme situations of severely depleted glutathione caused by chemical exposures, mercury or what have you, typical physiological levels of vitamin B12 may be insufficient to ensure survival of this vitamin. So just because the lab on your patient comes back and it says serum cobalamin is fine, you do not know if that's methylated cobalamin or cobalamin 2 or 1. You have no idea what it is. I'm trying to work with labs to be able to determine this so we can get a snapshot and say, look, your B12 is oxidized, you need to have fresh cobalamin levels. So if you see an elevated cobalamin on a patient's lab, doesn't mean they don't need it. It means it could be all oxidized and it's not able to do work. It could mean a bunch of other things as well. So this is a fantastic paper to read as well. A new role of glutathione protecting B12. And we showed you why that is, right? Hydrogen peroxide will shut down methionine synthase reductase enzyme. Yes? It's a combination of everything. It's the SAM, the B12, the SAM, the riboflavin. I don't know if it's the flavodoxin specifically, but I do know that I looked at the cofactors for methionine synthase reductase, and it was those three, SAM, B1, and B2. Yeah, and a key point about FAD, FAD is vitamin B2, but it's activated. So patients who are hypothyroid, T4 converts inactive vitamin B2 to active vitamin B2. So if your patient is hypothyroid and you're just giving them T3 and you're not supporting their T4 levels, their ability to produce FAD goes down. And FAD is needed to recycle glutathione. It's needed to produce your FAD for MTR recycling as well. It's in multiple enzymes. So this is a big picture. So what I do is I look at big picture things which all clinicians need to understand, and you need to understand the biochemistry of it, because hypothyroidism makes our patients tired, and makes their hair fall out and their skin dry and all these other things, but it also does things like it doesn't activate vitamin B2, which is super important to understand. So again, hydrogen peroxide, if you have too much and you have too little glutathione, here's your glutathione down here. If you have too little glutathione, this increases in amounts and it keeps this cobalamin oxidized, which then shuts down your methionine synthase. We've said that multiple times. So MTHFR, once again, since I am known as the MTHFR guy and people that think that's all I do, but I guess it makes sense since I have a website dedicated to it, but I do way more than MTHFR. So one copy of 677 or 1298 accounts for 12 to 57% of the population. So it depends on the ethnicity of the patient. So researchers reported a patient who received nitrous twice within a period of 10 weeks who developed some myelopathy and macrocytic anemia. And then they did some further evaluation later on. They found elevated homocysteine, low B12, and MTHFR 677 present. It resolved with folate and B12. And I wish they would not just say folate. At least they said folate, not folic acid, so that's good. But it would resolve a lot faster with methylated folate and methylated B12, and I will show you proof with the methylated B12 here soon. And so they're saying that although screening for all patients for elevations of homocysteine doesn't seem to be relevant, and it seems to be a pain in the butt, and it's impractical, given the situations and the prevalence of MTHFR, and it is increasing in the population, and I will get into that later as well, MTHFR is. So I think screening is very important. So screening for homocysteine, dentistry, especially biological dentistry, is already very expensive, but so is irreversible nerve damage or dementia. And then you have to remember too, family members. The whole family comes to see you usually, right? So if it's the mom, it's the dad, it's the kids, it's the grandparents, it's everybody. So it's genetic, so it's passed on through. So that's something to be aware too. And this is a very significant paper. Again, I don't want to read it, but this is talking about an elderly gentleman who underwent surgery, had nitrous oxide, and he basically woke up, forgot who he was, where he was, and he later found out that he had very low B12, had macrocytosis, macrocytic anemias, and they identified that and they gave him the B12 and everything luckily reversed. But if you catch it too late, what happens? Irreversible nerve damage, right? So Tylenol. How many of you guys give Tylenol for post-nitrous treatments to relieve pain at home? Raise your hands. Okay, a few of you. Okay, so is it bad? This is a great paper to read about and what it's doing to our patients. One dose of Tylenol, one standard dose of Tylenol can deplete your patient's sulfation pathway completely, one dose. Autistic kids' sulfation, their ability to work with the sulfation pathway is already greatly, greatly diminished. So if you give Tylenol, you're further depleting that. There's an interesting paper that was written by William Shaw that looked at the prevalence of autism in vaccinated kids in Cuba and the US. Cuba has a very similar or identical vaccination schedule as the United States. The only difference, in the US, we frequently give them Tylenol because of the fever or pain. In Cuba, they don't. Cuba, the rate of autism is very, very low. In the US, it's very, very high. So they say, well, maybe it's associated between not only giving the vaccinations, but also giving the Tylenol, depleting their sulfation pathway. So it's very interesting, and they talked about multiple routes of elimination and exactly what's going on with Tylenol, but detoxified by glutathione partially. So N-acetylcysteine IV is the preferred method of treatment that's utilized in the ER. So the key points is to remember all the biochemistry I just showed you. No. The key point is to understand that nitrous oxide is going to have an impact on all your patients to some degree. Whether or not that degree is visibly seen in their symptoms, it doesn't matter. Biochemically, you have altered their methylation. And I believe in optimizing our patients. You guys believe in the same thing because you guys are biological dentists. You guys are the leading dentists in the world, and there's only a handful of you, which is really, really shocking. Okay? And I've talked with Hal Huggins multiple times. My wife's root canal's gone, mercury amalgam's gone, and so I'm totally on board with what you guys do. Okay? And I recommend you guys all the time. So we have to optimize our patients, and understanding what this does biochemically will put you guys even higher in the driver's seat of optimizing your patient, because you're already doing that by using less harmful materials. And sometimes you do need to use nitrous oxide, but if you know how to bypass it, which we're going to get into in the next hour and a half, is very useful. And delayed negative effects. So just because you don't see it doesn't mean there hasn't been a problem. So it can be delayed. That's important. And try to find other ways to reduce anxiety. If the number one reason to utilize nitrous is to reduce anxiety, then let's try to come up with other things and give you tools to do that. I'm a naturopathic physician. I'm labeled as a quack. We have some quackery things to reduce anxiety. Okay? Team care is super important. I'm all about integrating medicine, MDs, NDs, DOs, DCs, nutritionists, doesn't matter. Ultimate goal is to improve our patient. Screening. You can screen with questionnaires. Are you taking antacids? Are you under tremendous amounts of stress? Are you a vegan? Are you vegetarian? All these things can immediately identify if your patient's at risk or not. Are you elderly? Because as you get older, your stomach acid levels drop and decline. As those decline, your B12 absorption also declines. So you need to inform your patients about the risks of nitrous oxide, and you need to support it, pre and post. And again, ending with epigenetics, and thank you. And to answer a gentleman's question, are you the person who asked the question to the prior doctor who presented before me about sulfation? Are you in the room? Okay, so sensitive to sulfur? Okay. A few reasons for that. Molybdenum is definitely an issue. Suox is tested by the 23andMe genetic test, so you can do that. If you get your raw data, you can order the MTHFR support report. That's mthfrsupport.com. You can download your raw data, run the report, and you can see if Suox is in there. Do know that sulfites deplete vitamin B1, thiamine. So if you're taking a lot of, or you're generating a lot of sulfites, or you're eating sulfite-containing foods, then do know that your B1 might be depleted. So that could be something to look at. Thiamine 50 milligrams once to twice a day could be something to look at as well. Sulfation pathway is highly specific to sulfur, but it's also very easily overwhelmed. So the backup pathway to sulfation, there's multiple SNPs in that pathway as well. Food coloring inhibits sulfation pathway, so don't eat any colored foods. And since sulfation is super important to get rid of dopamine, you want to tell your patients who are children and the parents of these children to not give food coloring. Don't give lollipops with food coloring after, right? Or everything, good job, here's a lollipop with food coloring, because food coloring inhibits sulfation. Sulfation is needed also to reduce dopamine levels, which then also relieves the pain. But also another thing to look at is glucuronidation. Glucuronidation is the backup pathway, the sulfation pathway. And glucuronidation, you can use calcium D-glucarate. That will really open that up. And there's other things which inhibit glucuronidation as well, various meds, and you can look at things that promote glucuronidation and inhibit as well. I've gotten into that in the past in prior conferences too. But I would give calcium D-glucarate a shot, B1, molybdenum, and avoiding things which affect your sulfation pathway. The great book that gets into the sulfonotransferases, which is the sulfation pathway, is Toxicology. It's a book literally about that thick, and it gets into serious details about sulfation pathway and things that inhibit it, and try to remove those things. Yeah.
Another question?
Question, you said screening questionnaire. Is there a way that we can get this screening questionnaire to ask our patients?
Yes,
Repeat the question, please, because
yeah. Yeah, so the question is the screening questionnaire for screening our patients, if they're at risk or not. Is there a way that we can get that? Yes, in the next hour and a half, I'm going to be teaching you guys how to support your patient pre-op, how to screen them, what conditions are susceptible, what to look for in your patients. And then once you do that, if I also give you alternatives to nitrous oxide, I also give you a pre-op, before they come into you, say a week or two or a month prior to come into you, how you support them nutritionally and supplementally, and then during treatment and post-treatment. And everything is written in the slides. Right now I haven't written anything out in detail, but it is all in the slides on how to do that, and we're going to be discussing that next hour and a half. But a booklet is very important. A handout, I think, would be a better term. I'd like to get it on one piece of paper so it's not overwhelming to you or the patient. Yeah.
Okay, so you guys are about to take a break again, but please give me just a moment here. The HDA, Roberta Glasser, someone thought that the 2014